would be a good thing to record. Okay, here we go. So the exam is half multiple choice, half free response. Really simple. You get minutes for each section. Multiple choice questions, usually they give 60 multiple choice questions in 90 minutes. Okay, if you have some sort of special needs built in, then that's already been built in for you, and that's for you if you have that. But the rest of you will have 90 minutes for 60 multiple choice questions. And then you get a break. You get like a 10-15 minute break. Go back in for the essays, the free response questions, and you get 90 minutes for those. That's really what I wanted to concentrate on today. But since I am talking about, you know, the overall timing of the exam, what you need to realize is that on the exam day, the exam starts at noon for everyone. It's not like you can just log in a little bit later and still get your 90 minutes. If you log in at 12:30, you've lost 30 minutes out of your 90 minutes. It will shut down on you at the same time for everybody. Okay, just so you know that. All right. You only have that. And another thing about this exam is it's not very forgiving. You can't go back to questions that you left blank because you wanted to consider them more. You've got to answer it and move on. Answer it and move on. Answer it and move on. You're not going to be allowed to go back to another question and edit your answer. So beware of that too. Okay, the free response questions. There are going to be 6 of them. If you look at, I don't know if you can see my arrow. Yeah, the first 2, Camille, I believe it is. I believe that they don't allow backtracking, Camille. But that's what they told us. That's what they told us. Okay. Okay, the first 2 essay questions are the long ones, the ones you want to budget most of your time for. You have 90 minutes. I would, yeah, I know. Don't. This is college. You're in college. The first two questions you should budget about 20 minutes each for those. Those are the ones that are worth the most points, and they're almost always going to be about some sort of a graph that they want you to interpret, or some kind of data or experimental design. Sometimes they'll give you a question and a situation and ask you to design an experiment, right? Or sometimes they'll give you a graph and experiment and ask you to analyze it, right? So that's generally what question 1 and 2 are about. The 4 questions that come after that are shorter. You want to budget maybe 10 minutes each of those. And you also have to consider that the first 10 minutes of those 90 minutes, the best strategy is just to look and read all the questions. Make sure you've read them all, and then go after the ones that hit you at first as being, oh, I know that one, right? If there's one that you feel like, oh, I know that, I'm really confident on that, go do that one first. and such. Okay. So these questions down here, 3, 4, 5, and 6, are going to be shorter questions. You don't want to spend as much time on them. And as always, with any of these FRQ questions, they're looking for quality rather than quantity. So they're not looking for flowery language. They're not looking for intro and conclusion paragraphs. They're just looking for, you said the term that they were looking for and you explained it correctly. That's it. You get the point. Okay, so let's go into a little bit more detail on that. All right, so for the longer ones, generally interpreting, evaluating experimental results. That's going to be what those first 2 are usually going to be. So they'll give you a description and data from an authentic scenario, and then ask you to, these are the words that they'll give you. They'll say describe and explain, identify, analyze, make a prediction, and justify it, right? Those tend to be the words that they'll use. They'll give you authentic scenarios that are going to look like Greek to you. There's going to be an experiment you've never heard of before, using organisms with scientific names you've never heard of before. They're going to try to intimidate you, okay? But every question that they're going to ask you is going to come back to basics. It really is. The best way to answer these questions and get them right is to read through and realize, oh, this looks complicated, but really all they're asking about here is protein synthesis. Or, oh, this looks complicated, but really all they're asking about here is the cell cycle and mitosis. That's all. Or, oh, this is just about enzyme function. So that's the kind of aha moment you're going to hope for each of these questions, right? Okay. Concept analysis, right? So the four little shorter questions are going to be, they'll give you shorter scenarios or maybe a little diagram or something or a picture, and then they'll ask you questions about it. So describe, explain, predict, justify tend to be the keywords that they'll ask you about there. So let's go into those keywords a little bit, because that's an awful lot to digest. So let's take it one at a time. Describe versus explain. Okay, describe means provide relevant characteristics. Explain goes into the how and why. What do those characteristics cause to happen? So right now, here's a picture of a mitochondrion, right? Describe how it's compartmentalized. I've got the picture for you. See if you can come up with some sentences. They don't have to be complex, okay? You can even use the words in the picture to help you. Describe the compartmentalization of a mitochondria. See what you can do with that. It's not asking you to explain what that compartmentalization will allow it to do. It's just asking you to describe it. Okay. Ah, that's beautiful. First one's really nice. Good. An inner and outer membrane that creates two compartments. Yeah. Two membranes. Yes, an outer and an inner. That's beautiful. That's beautiful. That makes two compartments. Yes. Or two sort of spaces or rooms within the one organelle. That's it. So what you definitely want to do is mention that there's an outer membrane and an inner membrane that create two chemical spaces. That's it. That's a good description. Looks like you guys have got that. Yes, that outer and inner membrane that creates two chemical compartments. Beautiful job. You got it. You got it, guys. All right. Let's go to the next one. Now, math keeps lighting. Okay, now go to the explain. This is a little harder. Explain why the compartmentalization of a mitochondrion is important to its success in making lots of ATP. Now here, you may have to really go back into your memory about aerobic respiration, right? Why were there two chemical compartments there? And why was that so key in churning out a bunch of ATP? See if you can remember that. And it's okay to make mistakes here, okay? It's perfectly fine. This is the right place to try, the right place to make mistakes. Surface area sort of has something to do with it, but it has more to do with the different chemistry on either side of the membrane. It has to do with the concentration. Yeah, there we go. Concentration gradient. That's it. You'd probably get most of the point from just saying because it creates a concentration gradient, because there's more of something on one side than there is on the other, right? And to really get the full points for it, you would mention that it's a hydrogen ion difference in concentration, right? So it allows the hydrogen ions to get really concentrated in the outer compartment, and then they have to flow back through ATP synthases. Remember those things? The hydrogen ions that flow back through the ATP synthases, that's how the ATP gets made. So having two compartments allows you to make a gradient for the hydrogen. And then the hydrogen is forced to go back through the ATP synthases, and that's what makes the ATP. So there's a bit of review there for respiration. We certainly aren't going astray with that. But yeah, what I'm seeing here is just about the right amount of writing to get those points for an explanation. That's pretty good, yes. The important keywords would be the creation of a concentration gradient, hydrogen ions, and ATP synthase. Those would be the keywords to use and to be able to explain what they have to do with each other, okay? Okay, so that's a pretty good experience there. But you see the difference between just simply describing the mitochondrion and then explaining its function and how the structure fits the function. Very, very common type of question. All right, good job, guys. Let's go on to the next one. Okay. Identify and calculate usually are the least amount of writing, right? The least. So for both identify or calculate, you would just be usually writing just a single sentence that just says, you know, the eukaryotic organelle that conducts the process of aerobic respiration is, and then blank, right? And calculation would involve, you probably just need to show your work. I would multiply this times this, and I would get this answer, and here's my units, right? Okay. So what I want you to do here is just give me the answer to this one, this first one here. What's the organelle that conducts aerobic respiration? Kind of a gimme. Right. Easy point, right? Easy point. Because I didn't give you any data for the calculate, so don't worry about that. Yep. Mitochondria, that's it. You got it. Don't worry about number 2. I just want to make sure that you make it as a full sentence. That's it. Good. That's it. All right. And it is a good idea to incorporate the language of the question in your answer to some degree, especially if they're asking you to identify, right? So just give it back to them and fill in the blank that they want. It also kind of ensures that you're actually answering what they want. One of the most important strategies for these FRQs is to make sure you understand the question. You know exactly what they're asking. Okay, let's try another one. Okay. Predict versus justify. Okay. When you're predicting, you're stating the causes or effects or a change in or disruption to one or more components in a relationship. Boy, does that sound like a clackety-clack definition. All right. If they ask you to predict something, actually predict something. A lot of students get the order to predict, and instead of predict, they start to explain, and they don't predict anything. So make sure that you give a prediction if they ask you for a prediction. So, and it doesn't, and usually with predictions, they'll take lots of different answers as long as you can justify it. Okay. So try this one. Predict what would occur if a mitochondrion is deprived of oxygen. Just write a prediction sentence. Don't justify anything yet. Okay. What do you think would happen? There are more than one correct answer here. Just do the answer to this. Predict what would occur if a mitochondrion is deprived of oxygen gas. You don't have to explain it. Just predict what you think would happen. You got it. Okay. All right. I think I like this better because this sticks more to the mitochondrion and doesn't move into the cytoplasm, right? It would produce less ATP or it would stop producing ATP. That would be a good answer. Or the ATP production would decrease. That's also a good answer. That is a prediction. You could also say that it would stop breaking down glucose, or it would stop breaking down pyruvate, right? Or you would get less carbon dioxide produced, would also be a valid answer because that's also a product, right? But I think lower production of ATP is probably the safest and best prediction there. That's good. That's a good one. Switching to cell might switch to glycolysis, but if they're asking about what happens to the mitochondrion, you want to stick to that. And it would produce less ATP. But I guess it would depend on how the question is worded. Okay. I can't remember if I wanted you to put the justification here or not. I think I put it on the next one. Hold on. Oh, no, I didn't. Okay, never mind. Okay. Okay, can you justify your prediction? This is good review anyway. See what you can do with that. Okay, less ATP produced because, why? Specifically, what does oxygen gas do for ATP production? That's a good question. Okay, this is good review. So remember that oxygen gas, remember that term final electron acceptor? Yeah, that's a really good term to remember. That's what oxygen does. Oxygen is the final electron acceptor. Even if you can't explain that, you'll probably get a point just for saying that. Okay? So that's why, because oxygen is needed to get the electrons out of the way so the electron transport chains can keep moving electrons. That's why. Those electrons move, and that is what's pumping the hydrogens out. And remember, the hydrogens create that concentration gradient that then causes the production of ATP through the ATP synthesis. Remember that? That was a long time ago, I know. That was a long time ago, but really, really good review, because aerobic respiration is a very basic process. Yes. Yes, oxygen, it accepts the electrons from the final electron transport chain protein and causes the rest of the electrons to keep moving through the electron transport chain. It keeps the electron transport chain going, which maintains the hydrogen ion gradient. Right. So those would be the key things that you would need, because oxygen is the final electron acceptor. That's one. Because it keeps electrons moving through the electron transport chain. That's two. And the ETC is what maintains the hydrogen gradient, which is pushing the ATP synthesis to phosphorylate ADP. Yeah, remember all that? Okay, that was a mouthful. All right, good. Okay, let's go to, let's do a real one now. Actually, I love this cartoon. Wonder where Einstein got his inspiration from. Okay. All right, just take a minute and look at this diagram. This is a very typical AP Bio diagram. Just take a look, read quietly to yourself for a minute. There's no question there, just read it. Okay. Can I go to the next page? All right. Here's part A. A skin cell completes one round of the cell cycle. Describe the products. So this may seem misleading, right? It's actually not really asking about that diagram. It's asking about a basic thing that you guys learned called the cell cycle, right? This is where having a pad of paper out and a pen to scribble things down is a really good idea. What was the cell cycle? When it goes around once, what comes out of it? And we're actually not asking about the level of molecules here. Not molecular products, but something else. Yeah, that's it. You guys are so clever. Yes. You get 2 daughter skin cells, exactly. Each with, oh, not with half the original DNA, but actually with the exact same, right? Unless your skin cells wouldn't be going through meiosis, right? So it would be the same. It would be the same, identical, right? 2 identical daughter cells that are going to be skin cells. That's it. That's it. That's all they wanted there. That's good. The cell cycle looks like this, remember? Remember that? So a skin cell completes one round, 2 diploid cells with identical sets of DNA. If you added the word diploid, bonus, right? If you just said identical sets of DNA, you've got it. Okay, try this one. Now, based on the figure, explain how P53, which is a protein, regulates the cell cycle in the presence of damaged DNA. So let the diagram guide you. Let the diagram guide you. Yeah, that's pretty good. It produces a protein that is an inhibitor to CDKs. That's it. It causes the production of a protein that inhibits a CDK. If you said that, you got it, because that's what the diagram shows you, right? P53 protein, if it detects that the DNA is damaged, it goes and wakes up a gene that creates a protein that inhibits CDKs. That's the correct explanation. Yep. Also yeah, you got it. Good. Codes for, yes, that I like that. That gene, or actually it's not a gene, but the protein activates the gene that codes for an inhibitor. That's it. That's right. And therefore, without the CDKs working, it would it stop or start mitosis? Remember what CDKs did when they were working well? They would promote mitosis, right? They would encourage cell division. So if you inhibit it, then essentially what you're doing is, well, okay, I'm not going to give you the answer because I want you to answer the next one. Okay, so the answer is P53 stimulates expression of the CDK inhibitor, preventing progression of the cell cycle. Okay, so I put it in simpler terms. You don't have to use all those fancy words. You could say, if DNA is damaged, P53 activates a gene over here that causes transcription and translation of a protein that inhibits CDKs, right? That doesn't use any language that you guys don't know. You can do that, right? Okay. How are you guys doing? Yeah, I think that's literally, okay, which one are you? You have no comments? No comments from the peanut gallery? Okay. Okay. All right. You definitely need a break. When we come back, I'm going to put you in groups and you're going to do a practice FRQ in a group. Okay? All right. Go and make yourself a sandwich. Put your toes in the grass, something. But definitely take a break.